Y'all, today we are going to start talking about basic experimental design and some elements and principles that have to do with experiments. We also will have a quick discussion here about what an observational study is, similar to an experiment, but also different. That's why similar, not the same. An observational study is where actually the key thing about an observational study is that we are not controlling anything. So we are just going to, I don't know if that's two L's, please correct my spelling. We are going to observe what is happening with subjects, or we can go back and look at previously collected data about a sample, and then we can take that information and draw conclusions based on those observations. And so you can see there are two types of observational studies. There are the prospective studies, where we select a group to study and then we sort of follow along with them, not literally, but we collect data periodically on them without controlling anything that's happening in their lives. A retrospective study is where we go back and look at information that's already been collected and draw some conclusions. So the things have already happened. We go back and look and then draw some conclusions. An experimental study is different in the way that we have control over certain variables. This word manipulation might sound negative, but we want to control the environment so that we can try to draw conclusions about specific characteristics or measures of interest without anything else, or hopefully without anything else influencing that. So here we have a measure of control, which takes us out of the observational study arena and into an experimental study. And this is the only way to show a cause-effect relationship. We can talk a lot about things being related or having some sort of association in this class, but the only way to show cause and effect is by doing an experiment. And of course, that experiment has to be done well. Part of doing that experiment well is making sure you have the elements of a good experiment. Number one is we want to have some control. So typically we'll have different groups in our experiment, and we will be controlling the variables. More on that later. We need randomness. When we're doing any sort of data collection, we want randomness involved so that we have representation. In sampling, randomness comes from how we select people to be in our sample, how we select elements to be in our sample. In an experiment, typically the people come to you, they sign up for the experiment. The randomness comes in the assignment. So in an experiment, we will assign people or animals or subjects to treatments, and that is done randomly. So everybody has an equal opportunity to be randomly assigned to any of the treatments in the experiment. And so that will hopefully mitigate some bias or make sure that we have a representative experiment. We need to be able to repeat. I probably should have said repeatable instead of replicable, especially because it's hard to say. We need to be able to replicate our experiment, and typically that means we are doing it on many subjects. So we are not, if we're testing a medicine, we are not going to have one person take the medicine. We are going to have many people take the medicine. And then good experiments typically have some element of blocking, which is not required, but it is helpful. And that is part of the way that we can control any sort of outside influence. And I will talk about more on this in a minute. One that I didn't write down here that I will talk about is blinding. Really good experiments usually have some level of blinding as well. So let's talk about the variables here. We are typically looking to see if one variable, that independent variable, has some sort of effect on a dependent variable. So in this scenario where we've had some cats and dogs sadly dying because the food was contaminated, the manufacturer who produced that food says that they've reworked it and they now have food that is safe, but now they have to test it, and so we need to run a trial. So the independent variable in this scenario is the food. So if we were to do an experiment here, we would be feeding our dogs and cats this new food. And then we'd also probably want to feed a group of dogs and cats some other food, maybe some other brand food, just to be able to compare the health of the dogs and cats after eating the new food versus a food that we already know is safe. So in this case, the outcome would be the health health, that's how you spell it, of our pets. We always want to be able to compare results from one group to another. So in this same scenario, we would probably have two groups. As I stated before, we would have a control group, and that control group would probably get some food that we already know is safe. And then we would have our treatment group, and that group would get our newly formulated food from this brand that was having the unfortunate deaths. We could then compare the health of the animals eating our new food to the animals eating the food that we already know and have determined was safe, to see if it in fact does provide a healthy meal for these animals. I mentioned blocking. We do not always have to block, but typically there are reasons why we would. And in this example, we would want to block out the pet type. So what we would end up doing is running the same experiment simultaneously, one with dogs and one with cats. So we would take all the dogs that were part of this experiment, and we would randomly assign some of the dogs to get the already known to be safe food, and some of the dogs to get the newly formulated food, and then we would compare those results. Then we would take some cats and randomly assign some of the cats to the food we know to be safe, and the other cats would go to the newly formulated food. Then we'd compare their results. We wouldn't necessarily want to compare the results of dogs eating these foods to cats eating these foods, because there could potentially be some issues still with either the dog food or the cat food, because they eat different types of foods. And so that would be a reason to block, so that we didn't get our results confused because the two different types of pets have something different happening. Blinding is also helpful to conduct a reliable experiment. We have single or double blind types of experiments. We have three groups. We have the group of subjects who are participating in the experiment. we have the group of individuals, sometimes doctors, maybe veterinarians in this case, who are administering the treatments. And then we have a group of people who are going over the results. And so if one of the groups, either the subjects or the people administering, if one of those groups doesn't know which treatment is being handed out, that is called a single blind. So let's see here. I'm going to pause for a moment. So if one of these three groups, specifically the subjects or the doctors or the people administering the treatments, if one of them does not know, so one does not know, I'm going to say one, no, nope, they don't know. If one of them doesn't know which treatment they're getting, then that's called a single blind experiment. For example, in this study, the pets don't know which type of food they're getting. They're just happy to have a meal. So if they're the only ones who don't know which type of food they're getting, that's a single blind experiment. If two of the groups, so the subjects or the doctors, the administrators of the treatments, if they both don't know, that's a double blind experiment. And so that in that case, only the researchers, only the people who would be looking and analyzing it, the data, would know who got what treatments. And double blind is the best way to go if you can. That's not always possible. And that's because sometimes, depending on what you're studying, maybe it's a new pharmaceutical, if you think you're getting the medicine that's supposed to help you, your mind can play tricks on you. That's called the placebo effect, where you think you're getting better because you know you're getting this medicine that's supposed to help you. Similarly, in some cases, the person administering the treatment could potentially act differently around the person they're giving the treatment to, knowing if they're getting the real treatment or if they're getting the control. And so in order to control that sort of like human nature element, we will blind our studies. One thing that we're always not able to mitigate out, what we're trying to get rid of, is something that's called confounding, something that's related to our variables that will have an influence without us being able to sort of separate that out or account for it. Here's an example where we have a weight loss study. So we are going to have one group do one type of exercise probably, and one group do a different type of exercise, and then measure the differences in weight loss. Well, we might have some of those participants actually start eating healthier at the same time. And if you've read any weight loss information, you'd know that exercise and eating healthy is the best way to go. And so without the researchers knowing, some people could be eating healthier, meaning they lose more weight, which will have an effect on our study. So if this is hard to read on the screen, this is also on Canvas, so you can read it there. But what I want you to do is read through this study and answer these 4 questions, and then we will discuss them at the beginning of next class. As always, if you have any questions about this lesson, please ask me either before you leave today or at the beginning of the next class.